Assalamu alaikum. Today we are studying cytokines. A cell can be activated by three ways, either by neurotransmitters or by hormones or by cytokines. Cytokines are the small signaling molecules. As you know, the immune cells are widely distributed in our body, like lymph, blood, and some other organs. The immune cells are communicate with each other. This communication is possible by cytokines. Cytokines are small soluble signaling molecules and act as immunomodulating agents. Now we discuss the characteristics of cytokines. Cytokines produced and secreted by wide variety of cells. They act as intercellular mediator. Cytokines bind to specific receptors on the surface of other cells, and these receptors are called cytokine receptors. Now we discuss the chemical nature of cytokines. Cytokines are chemically protein or glycoprotein as they cannot cross the lipid bilayer of cell membrane. That's why they attach on their specific receptors. Their molecular mass are less than 30 kilodalton. They have short half-life. They are effective at low concentration because of specific high affinity cytokine receptors. Now we discuss the mode of action of cytokine. Once cytokines attach on the cell surface receptors, they alter the activity of the cell in three ways. Either a cell may prepare to divide, or undergo growth and differentiation, or secrete its own cytokines. There are three types of mode of action of cytokines. Number one, autocrine. Number two, paracrine. Number three, endocrine. Autocrine means cytokines secreted by cells and act on its own surface, like this diagram. Paracrine means secreted by one cell and act on nearby cell. And endocrine mean cytokines act like hormones secreted by one cell and act on the other cell which is distant from it. Cytokines travels by systemically. There are five types of cytokines exist in our human body as well as the other vertebrates. Number one, interleukins. Number two, interferons. Number three, tumor necrosis factor. Number four, colony stimulating factor. And number five, chemokines. Now we discuss one by one. Interferons. Interleukins. Interleukins are a group of cytokines that are secreted by white blood cells as well as other body cells. They are of interleukin 1 to interleukin 35. Interleukins play main function in immune system. They are secreted by macrophages, T lymphocytes, monocytes, and endothelial cells. For example, We discussed this diagram. For example, macrophages secreted interleukin 1. Then interleukin 1 act on the helping T cells on its receptors. So the helping T cells activated and secreted interleukin 2, interleukin 4 and interleukin 5. These interleukins act and attach on the B cells on its receptors. Then B cells differentiated into the plasma cells and then plasma cells secrete the antibodies on the against the specific receptors and the specific antigens. So they promote the T and B lymphocytes development and differentiation and hematopoietic cells. The hematopoietic cells are present in our bone marrow. Interferons. Interferons are a group of cytokines that made and released by host cells in response to the presence of several viruses. There are two types, type 1 and type 2. Type 1 interferons include the interferon alpha and the interferon beta. The type 2 includes the interferon gamma. Now we are discuss one by one. Interferon alpha produced by viruses or viruses infected host cell and suppresses by interleukin 10. Interferon gamma produced by helper T cell and natural killer cell and activated by interleukin 12. Interferon beta produced by connective tissue and fibroblasts. Now we discuss this diagram. For example, when a virus attack on our body cell, the body cell produce the interferon alpha because the nucleus is activated and synthesize the interferon alpha. This interferon alpha released and attach on the nearby cells on its receptors. Then these cells produce two types of substances. One is the protein kinase and the second one is ribonuclease. The ribonuclease ingest and digest and catalyze the virus RNA or DNA, while as the protein kinase catalyze the protein coat of the virus. Furthermore, the interferon alpha act on the macrophages on and attach on the macrophage on its receptors. Then macrophages activated and produce the interleukin 1. Then interleukin 1 attach on the helper T cells. The helper T cells activated and produce and release the interleukin 2, interleukin 4 and interleukin 5. Then these interleukins attach on the B cells, then B cells differentiated into the plasma cells and produce the antibodies against these viruses. Tumor necrosis factors. Tumor necrosis factors are produced by mast cells, macrophages, and T cells. They are firmly anchored into the cell membrane and regulate immune response and inflammation. So they are transmembrane proteins. For example, macrophages secrete TNF alpha and it binds to the neutrophils on its receptors. So neutrophils are activated and ingest the pathogens. Like this diagram, macrophages release the tumor necrosis factor alpha. Then tumor necrosis factor alpha attach on the neutrophils on its receptors. Neutrophils activated and come inside the site of infection and ingest the virus or bacteria. Colony stimulating factor. These are essential for the growth and differentiation of thrombocytes, granulocytes, and lymphocytes. Furthermore, they ensure that the body is supplied sufficient number of white blood cells of all types. Thus, it will determine the immunity. Now, we discuss the types of colony stimulating factors. There are three types. Number one, monocyte colony stimulating factor. These are produced and released by T cells and bone marrow and affect the growth of monocyte. Number two, granulocyte monocyte colony stimulating factor. These are produced by T cells and macrophages and they help in growth and differentiation of dendritic cells. Granulocyte colony stimulating factor. These are produced by monocyte and they help in differentiation and development of neutrophils. Chemokines. Chemokines are small protein cytokines. Their molecular mass is 8 to 10 kilodalton. Chemokines induce the chemotaxis process. They activate G protein couple receptor and migrate through concentration gradient. Chemokines are produced from immune cells like T cells, macrophages, dendritic cells, and B cells, and non immune cells like endothelial and epithelial cells. The main function of chemokines is to attract the cells. Chemokines control the movement of leukocytes. They attract and give signal to leukocyte to move. For example, if there is an infection, the chemokines secreted at the site of infection and give signal to neutrophils to migrate at the site of infection and ingest the damaged and infected tissue. Now, this is all about the cytokines. Please subscribe my channel for more biological videos and like and share. Thank you very much.